In fact, it's really cheap to get your entire genome sequenced. The real question is, what do you do with all the extraneous genes you don't know anything about? So these are the ones commonly associated with cancer. So there are specific recommendations. So she has what's known as a Scottish mutation in CDKN2A. And as you remember, this is hypothetical, created. So allows me to sort of go through some of this. I'll quickly go through because this is the most common mutation you're likely to confront in your practice. What are the functions of the genes? So first, this is a complex pathway, but I've isolated and highlighted the single protein that's important in this pathway, which is CDK4. So the gene is P16ARF, shown there in the gray. And P16 inhibits the CDK4, and that's critical because the CDK4 goes unarrested or unchecked. It phosphorylates a series of proteins, including the retinoblastoma protein, which then triggers cell cycling. So that's why they start getting cancers. The other thing about this gene that's critical is there's a protein called ARF, or P14ARF, and that, in essence, sort of guards the P53 protein, all right? So when CDKN2A is absent, P53, which is the guardian of our whole genome, is destroyed. So in a single mutation, you can create increased birth of cells, and you're diminishing the death of apoptosis, or the death of cells. So because of that, patients with CDKN2A mutations are at increased risk for both pancreatic cancer and melanoma.